This video is about the cell cycle. So it's going to be about chapter 12, and it's going to be explaining how we're interested in this point in the cell cycle. This is the point that is most commonly regulated. And chapter 11 is about cell signaling, cell communication, and regulation. Regulation as a kind of information, as a switch. And here's the switch, and it's at this G1 to S checkpoint. And so we're going to jump into the cell cycle, and we're really interested in this control point because this is the pathway that PDGF, the PDGF receptor, and its kinase activity lead to activating, stimulating, increasing the rate of cell cycle during wound healing. And so the PDGF is released from platelets that are in the area, stimulated because there's a wound like that. And so here we are zooming back, and the first thing to point out is that the book has the wrong location for this start control point, and it is here. It's right before S, and it's not a checkpoint. It would be called, it's called a control point. It's called start, or the G1 to S transition. Okay, and so here's our first look out at the cell cycle. And the first thing to say about the cell cycle is this is something that's depicted that's happening inside of cells, and it's depicted similar to, let's see if I have a figure of this. The cell cycle is similar to like the series of steps a washing machine goes through. So you start the wash, it fills up, and it suds the soap around with the clothes, and then it spins it out, then it rinses it, and it spins it out and spins it dry. So there's a series of cycles, and then it repeats. And so that's the name. The name cell cycle is akin to a washing machine cycle, where the process is a series of steps that are then repeated. And the cell cycle control is similar to the wiring and switches in a washing machine and how the washing machine is regulated. The cell cycle is happening inside the cell, mostly regulated by proteins that are floating in the cytoplasm of every eukaryotic cell. And I mentioned in one video that scientists who studied yeast won the Nobel Prize because these proteins that regulate the cell cycle can be found as orthologs in all eukaryotes. In other words, this cell cycle control is ancient, and the tools that are used to regulate it, the proteins that regulate it, have been used for hundreds of millions of years without changing very much. So we can study simple organisms and learn about humans and human health. Okay, so people have studied this cell division process for 150 years because they could see it under a microscope. And the first thing to say is, so historically, the cell cycle was studied under a microscope. And what you could see under a microscope is that nothing special happens during this whole period. And this whole period, nothing special appears to happen visually. The cell grows some, right? But it just looks like nothing special is happening. And this is called interphase. And then, quite abruptly, it usually takes about an hour, the cell goes through mitosis, where the chromosomes appear in the nucleus, and then the nucleus breaks down typically in most species, and the chromosomes line up and are separated, and two cells are formed. And so that's called mitosis. That's that period. So we have a long interphase and then mitosis. Cells that aren't going to divide again, like cells in our brain, are in interphase. Some cells, like in an embryo, there's a high rate of mitosis because the interphase is short. And the cells are growing fast because the interphase is shortened. So now modern biochemists and geneticists have found the details that happen within interphase, and they've divided interphase into three parts: G1, S, and G2. And the G stand for actually gap. And it's a gap between mitosis and S stands for synthesis. And synthesis S phase is when the DNA gets copied or replicated. So before the cell could divide, the DNA has to get copied so each cell can get the DNA. And so that's an essential part, but it's invisible during interphase. During interphase, the chromosomes are invisible in the nucleus, and it's not obvious when the DNA gets copied. The gap phases are important because this is really when the cell does its living. So when we talk about the cell's activity and the physiology and development of the cell, that happens during these gap periods. So that's the important part, and the S phase and M phase are essential to the process of cell division, like that. Okay, so G1 ends with the start of S, and the start or control point that is most often regulated in eukaryotic cells like people, our cells, is right at this transition. And so when cells go through start, they immediately start copying their chromosomes. And if you look at what the target of the PDGF stimulation is, it is proteins that regulate DNA synthesis. So if you get the cells to start regulating those proteins and stimulate that replication, boom, it goes through synthesis like that. Okay, so to erase this, there are three especially famous points in the cell cycle where regulation takes place. This one, and this is one we're especially interested in, and then a checkpoint that's different than a control point, a checkpoint right before mitosis. And a checkpoint is when the cell checks to see how things are going. And in a sense, a control point is sensitive to external factors, and a checkpoint is sensitive to internal factors, things happen inside the cell. The G2 checkpoint is checking for, to make sure that the DNA replication is complete. So you don't want to divide the cell before you finish S phase. And it looks like there's a big gap here, but you want to confirm that. That would be a big mistake to divide the cells before replication is complete. Also, the checkpoint checks to make sure there is no DNA damage. If the DNA is damaged, there's an opportunity to repair it before the cells divide. So that's an important checkpoint. And then there's an M phase checkpoint, and that M phase checkpoint makes sure that the chromosomes are separated. So we want to get those fairly separated. And those are three famous points in the eukaryotic cell cycle that our book talks about. And the book goes on to talk about this one in detail. And the reason is because this one that we're most interested in, it's kind of complicated, but features of it, principles of it, are very similar to this one. So we're going to do that too. We're interested in this, and this is the bottom line. This PDGF stimulates the skin cell cycle here, but we're going to learn about the G2 checkpoint in order to understand features of the cell cycle. And where we're going to be is we're going to be inside any eukaryotic cell, and basically mostly in the cytoplasm with these regulatory proteins that regulate the cell cycle. Okay, so just another, this is this image of showing interphase. So from the book talks about this as well. And the book has a good coverage of this too, so hopefully the lecture and the book are complementary. Okay, so with this person, she's going through that start too frequently and pushing this button repeatedly like that. Boom, boom, boom. Do a load of laundry and just immediately start doing it again. Okay, so some replication here. So now we're going to zoom in and look at this. And this is the G2 checkpoint, like that G2 to M transition. And we want to stop and make sure the DNA is replicated and not damaged. Here's the book showing that. There's the cell cycle. There's the G2, and again, the book doesn't show these as being sharp separations. When the cell passes through the G2 checkpoint, it's in M phase, boom, like that. And that's actually an important principle, that when these guys go through those control points and checkpoints, they are in the next phase. It's part of the sort of definition of passing through this is that you are now in the next phase. Okay, so now the book introduces regulatory proteins that we'll talk about as well. Regulatory proteins that control the cell cycle. And an important one is this one, and it's called CDK, and it's a protein, So like all the other proteins in our cell, you know, it's folded up in a certain way, tertiary structure, and it has a job to do based on its structure that gives its function. And this is a cyclin-dependent kinase. So we saw that our PDGF receptor is a kinase, and we also saw a Wikipedia page that says there are 500 different kinases in humans. So here's a second one, cyclin-dependent kinase. The PDGF receptor kinase has certain target proteins. This has a different set of target proteins, like that. So it's going to phosphorylate a certain set of target proteins, turn some on, turn some off, depending on the structure of the protein target. Okay, and so then it shows this. This CDK protein it shows is present all the time. All right, so that's a key thing. It's present all the time, and it's going to get turned on here. And when it's turned on, it's referred to as MPF, you know, and that's called mitosis promoting factor, because it is the enzyme that throws the switch. Again, we're interested in information and switches, choices that cells are making, and here's a big regulator of a switch. When it's on, the cell goes into mitosis. Now, this protein must be important to be regulated, because if the cell goes into mitosis, when the DNA is damaged, the cell is going to be in trouble, like that. So this should not get turned on until the checkpoint has been checked and DNA is ready. Okay, so one way that this is regulated is because it depends on this cyclin protein. So MPF is composed of CDK and cyclin. And the cyclin is only made during G2. It starts to build up during G2. So that's one way that this CDK is regulated. CDK is regulated because it needs cyclin. And there is a second way that we're going to talk about that CDK is regulated. So CDK regulation, and this is actually CDK1. There are several CDKs in the cell, and CDK1 works on the G2 to M checkpoint. CDK1 is regulated by cyclin, and 2, it is regulated by removing inhibitory phosphates. So, how can CDK be present all the time and the cell's not in M phase? The answer is there's two reasons, basic reasons. One is it needs cyclin to be activated, and 2, it has inhibitory phosphates on it. So this protein is the target of kinases, and the kinases act here. So if we draw CDK1, let me just do this. If we put phosphates on here, a phosphate is added here. It's actually 2 phosphates are added here that are inhibitory phosphates that help turn off CDK. So see, it's an important protein to regulate. It's regulated 2 ways. Cyclin's degraded, that helps turn it off. Phosphates are added, it's turned off. So as long as those phosphates are on there, this protein will be off. And right here, it's off. And right here, the phosphates are removed. And then they're put back on here. So there must be kinase or kinases that function here to terminate mitosis. And there must be something that removes a phosphate here to activate it. And when enzyme removes a phosphate, that enzyme is called a phosphatase. So we have kinases that add phosphates to target proteins, and phosphatases that remove them. And just about every kinase has an alternative phosphatase. So kinase adds it, phosphatase removes it. And so there's a phosphatase that removes these kinases here. And this phosphatase that removes kinases is called CDC25. Like that. So there are 2 major ways that this MPF is regulated. And when they are activated, the cell will go into mitosis. And I made a handout, and I posted that on the handout website. here it is, but just to kind of emphasize that this is a handout I made, and I made this handout to try to show you a little bit more about activation of the CDK1 at the G2 to M transition. And the way this handout works is we start at time 0, the cell divides, mitosis, and we work backwards in time. And so the cell divides, and what happened before that? What happened before that is that MPF is active, and so the cell enters mitosis. MPF is made up of CDK1 kinase. This is an old name for it, CDC2. Why do these proteins have multiple names? They're studied in multiple species like that, and so sometimes different scientists discover the same comparable protein, ortholog protein, and have already given it different names. So historically it was called CDC2, and I knew it best then, so that's why I originally used it, but this is a better term, CDK1. Our author just refers to it as CDK, but CDK1, we have CDK1, 3, 4, 5, 6, like that. And so why does it enter mitosis? Because CDK1 is activated and phosphorylates target proteins. And so that's what happens to make the cell go into mitosis. Now, how did MPF get activated? MPF gets activated here at -2. It says CDC25 phosphatase, I just talked about that, removes 2 inhibitory phosphates at amino acid positions T14 and Y15 from CDK1, and this helps activate MPF. So if you look here, there would be 2 phosphates here, and they're back here. And let's just jump back here for a second. It says here, WEE1 and MYT1 are kinases that inactivate MPF by adding phosphates to CDK1. And when are WEE1 and MYT1 active? And the answer is this occurs at the end of the previous mitosis. So if we erase this just for a second and look back here, this is where WEE1 and MYT1 are active. And what they do is they're kinases, and one of their target proteins is CDK1, and they add the phosphates that inhibit CDK1 during interphase. CDK1 is present during interphase, but it is inactive. See? Boom, like that. Who had activated it? WEE1 and MYT1. Okay, so just to go to here again, so the cell divides. Prior to that, MPF is active and is phosphorylating target proteins. We'll talk about a couple of the target proteins that lead into mitosis later. And why did it get activated? How did it get activated? Well, one reason is that the phosphates were removed at these inhibitory positions by CDC25 phosphatase. Also, as described in the book, cyclin is added. This is cyclin-dependent kinase. So cyclin is added here also in G2 to make this thing active and drive it forward. Okay, now here's a switch or information control. What if the DNA is damaged? If the DNA is damaged, we do not want to go into mitosis because there's an opportunity to repair the damage. Like if your DNA gets damaged by UV light at the beach, before your skin cells divide, there's an opportunity for a DNA repair activity to go. And we'll learn a little bit about that in chapter 16, I think, about repairing DNA. So what if the DNA is damaged and you want to block the G2 to M transition? Well, you should keep this inactive, and one way to keep it inactive would be to inhibit CDC25. So let's go up here to -3. If the DNA gets damaged by ultraviolet radiation or radioactivity, for example, the cell should not divide until the damage gets corrected. The CHEK1 kinase, so this is another protein, CHEK1 kinase, gets phosphorylated as a result of that damage and is activated if the DNA is damaged. So CHEK1 could be, we can draw it up here, CHEK1's not phosphorylated, and the DNA damage leads to phosphorylation by yet another kinase, and we won't learn about that one. So CHEK1 is phosphorylated, it's active, and when CHEK1 is active, it is a kinase, active check 1 is a kinase that phosphorylates CDC25 and inactivates it until the damage is fixed. So if we want to block entry into mitosis, let's keep CDK1 inactive by inhibiting CDC25 by phosphorylating it. So, and when CDC25 is phosphorylated, it's interesting just to show you this idea that that the active MPF is inside the nucleus, and so CDC25 needs to go into the nucleus to remove those phosphates, and CDC25, when it is inhibited during DNA damage, is held outside of the nucleus. So this protein here, 14-3-3, is another protein that helps keeps the CDC25 inactive and away from CDK1. So this is just a nice example of how there's extra important checks and balances. We really don't want to go into mitosis, so let's block it there. And let's block it there by inhibiting the enzyme and also putting the enzyme far away. Okay, and then just to go back here, this takes us back to that previous mitosis, and I actually already talked some about that. Some students will ask about this other phosphate, and this other phosphate is just some more, and I just added this to show you that there's more details to this story. This is a story of a high degree of regulation, high degree of controlling this switch choice, and we don't need to know about these proteins except for that there are more proteins controlling this pathway. And then remember I said we're really interested in this switch control point, but there are even more switch controls regulating this point. So that's why we skip down here, because this one is relatively simpler. That being said, in our handout we see it's still pretty complicated, but this is a nice way of seeing this control. This is like the wiring in a washing machine. I mean, if you open up a washing machine, it's just not that complicated. And these are individual proteins that in general are floating in the cytoplasm, or in some cases in the nucleus, of every eukaryotic cell. So you can look out your window right now, and you can look at the tree, and the tree has orthologs of all these proteins in every one of its cells, controlling the cell cycle. And you might see an insect fly by. That's got it, right? You might see a squirrel in a tree. Every single eukaryote, plants, animals, big and little, has the same story. So it's a profound story, and it has tremendous significance for our health. So when you boil down what are the most important stories in introductory biology, this is one of them. Again, this is a handout. The book talks about this same story, but it's hard to visualize. It's hard to comprehend it, and so I wanted to show it another way to try to add it. And so we've been here at this control, at this checkpoint, the G2 to M transition, and that's when CDK1 regulates its story. And you'll see, now that we know about CDK1, we're going to talk about it some as we go through the rest of our course. And that's that.